Hello everyone. Today's lecture is on structure based drug design, which is also abbreviated as SBDD. This is a very important topic for CSIR NET Life Sciences, falls under the Bioinformatics unit. In this video, we will cover everything you need to understand, like what is SBDD, how it works, what is molecular docking, what is binding site analysis, what is drug discovery pipeline, and what is docking. So, let us begin. First of all, we should know what is drug design. So, before moving into the structure based drug design, we should know the basics of drug design. So, in drug design, there are two main players. One is the target, and the another one is the drug, which is also known as ligand. So, the target is usually a protein that is present in our body. It can be uh, an enzyme, a receptor, or ion channel that is involved in a disease. For example, in cancer, various enzymes are overactive. So, target could be anything. On the other hand, drug is a small molecule that we design to interact with its target, like a key in a lock, where it modulates the target's activity to treat a disease. So, in drug design, the drug must bind to the target at the right place with the right shape and the charge to be effective. So, there are the two main approaches for drug designing. One is structure based drug design, which is our today's topic. We know the 3D structure of the target protein in structure based drug design, and then we study its binding site and a drug that should be best or fits perfectly in the binding site. So next is the ligand based drug design. The difference between the structure based and ligand based is the 3D structure, because in the ligand based structure is not available. Here the 3D structure is not known. We study already known active molecules and design the similar ones. So today we will focus entirely on the structure based drug design. So what exactly is structure based drug design? It uses the experimentally determined and computationally predicted 3D structure of a biological target to design, optimize, and evaluate potential drug candidates. The most important requirement for structure based drug design is the 3D structure. So, the 3D structure of a protein is a very much important here. So, we need to know the 3D structure of a protein. This structure can come from different techniques like the X-ray crystallography, which is one of the mostly commonly used structure prediction or commonly technique to give a very accurate protein structure. Next is the NMR spectroscopy, used for the smaller proteins in solution. Another one is the cryo-electron microscopy, which is used for the larger protein complexes. Or we can also predict the 3D structure of a protein by using computational methods or the homology modeling. So this is the method which we use to identify or to predict the 3D structure of a protein when there is no crystal structure available. So once we have the 3D structure, we can identify, the next step is to identify the binding pocket of a protein. So this is a groove or a cavity in a protein surface where the drug molecule is designed to fit perfectly. So this is the fundamental concept of structure based drug design. So now let us look at the complete step-by-step process or the workflow of structure-based drug design. There are 8 major steps. First step is the target identification. We know that target could be a protein enzyme or ion channel. So, in this step, first of all, we identify the protein, which protein is involved in the disease. The protein must be druggable, meaning it must have a suitable pocket where a drug can bind. So, in the target identification, we identify a protein with a suitable binding pocket. Second step is the structure determination, which means we get the 3D structure of a protein from a database called PDB. This is done using different techniques like the X-ray crystallography, NMR, cryo-EM, or by computational method like homology modeling. So these methods are used to identify the 3D structure of a protein, which are then further submitted into a PDB's database, which is a 3D protein structure database. Next, the third step is the binding site analysis, where we identify where on the protein surface the drug should bind. So this could be the active site or the allosteric site, the main functional site or the allosteric site, which we can call binding site. So these two can be the binding site of the proteins. So next step is the ligand preparation or the library preparation. We prepare a library of small molecules, and these small molecules are drug-like molecules. This involves adding hydrogen atoms, assigning charge to them, and generating different conformation or the 3D shapes in the ligand preparation step. Next step is the molecular docking. This is one of the most important step where we dock each small molecule into the binding site and score each pose and to identify the best fit molecule. So next step is the ADMET analysis, where we check if the molecule are safe or effective. So ADMET stands for absorption, distribution, metabolism, excretion, and toxicity. The seventh step is the lead optimization. So we modify the best molecule to make it even better, to so that we can reduce the toxicity effect of the lead molecule and to increase the selectivity also. So the last step is the experimental validation. Finally, we test in the lab in animals and perform clinical trials of the drug-like molecule. So now let us understand the binding site. As we earlier said, the binding sites are important sites because these are the site where the drug-like molecule can bind. So there are two different types of binding site. First is the active site. We have the active site, which is also known as orthosteric site. This is where the protein natural substrate normally binds. So let's say here is the this is the protein and this is the active site. It is directly involved in the enzyme's activity. Most drug targets this active site. For example, in HIV, the HIV drug Lipinavir blocks the active site of the HIV protease enzyme. So that's why active sites are the site which plays a very important role in binding. The second is the allosteric site. So these are not the active site, but they located away from the active site. When a drug binds here, it causes change in the protein conformation, which then indirectly blocks the active site. Allosteric drugs are important because they can be more selective. So here we have an example, the allosteric inhibitors of kinases. So next, now How do we find these binding site? It is important to find out this binding site. So we use different softwares like the site map. Next is the cast p, f pocket, and dog site scorer. These are the different binding site identification tools. Now the drug is held in the binding site by different forces. So the forces which are involved are hydrogen bonds, hydrophobic interactions, van der Waal interactions, electrostatic interactions, and pi-pi stocking interaction between aromatic ring. So now we got to know the different binding sites present on the protein surface, and these are important because they play important role in the binding of the drug like molecule with the protein. Now we come to the most important concept of structure based drug designing, which is the molecular docking. Molecular docking is a computational method that predicts the preferred binding orientation, or we can call pose, of a small molecule or ligand within the binding site of a target protein, and scores how well it fits. Think of it like a lock and a key. The protein in the lock with a keyhole, which is the binding pocket. The drug is a key. Docking predicts which shape of the key best into the lock. So, molecular docking has two components basically. So, the component one is the sampling. The computer explores all possible orientations and shapes of the drug like molecule inside the pocket. The drug can rotate, translate, and change its shape. This is the search step. Second component is the scoring. Once we have different poses, the computer scores each one. Lower score means more negative and the stronger the binding activity, whereas the higher the score, less negative, lesser the binding. This tells us which pose is good. So there are different types of scoring functions which can be used in molecular docking, like the force field based scoring function using physics equation like amber charm, and empirical function is the another scoring function. We have knowledge based scoring functions and the mla based scoring function. So next is, now we come to know a very important concept is the docking. As we know that docking is of different type, so here we can see the hard docking. What is hard docking? So look at this animation here. You can see the animation. So hard docking is also called rigid docking. So here the ligand is also kept rigid and protein is also kept completely rigid. No flexibility allowed in the hard docking. No means no part of it moves at all. The drug molecule also keeps its exact shape and it approaches the binding site. This is the lock and key model, just like a physical lock and key. Both are rigid here. This is fast to compute, and but in reality the proteins are never that much rigid in real state. Next we have the soft docking. So we can see here in the video the soft docking, because in the soft docking we call it flexible docking as well. Here we can see that the protein side chains are moving as the drug comes in. The pocket is actually reshaping itself to welcome the drug like molecule. Here, the protein side chains are flexible and ligand can change its conformation as well. This is the induced fit model. Now, let us understand the induced fit effect. This is the biological basis for why soft docking is much more realistic than hard docking. So there is a concept induced fit theory which is given by Koshland in 1958. It states that the protein shape is not fixed. When a drug molecule approaches the binding site, the protein's amino acid side chain move and reorganize to better accommodate the drug. It is like a handshake. Both hands adjust to fit together. So look at the diagram here. In step A, the protein's binding pocket is open and has its own shape. In step B, we can see here we have a drug-like molecule. This is a drug-like molecule. The drug approaches and start to enter. In step C, the protein's shape has changed. It has adjusted around the drug. This is called induced fit effect. Now, compare this with the older lock and key model given by the Fisher from 1984. So, in the lock and key model says protein is rigid, drug must be exactly the right shape so that it can fit inside the pocket. So there is no flexibility, and this model is used in the hard docking. On the other hand, we have induced fit model, which says that both the proteins and the ligand are flexible and drug can adjust. So in hard docking, we assume lock and key model. The protein is rigid, and on the other hand, in the soft docking, we use induced fit model, so protein can move. This is why soft docking is more realistic than the hard docking and gives the better results. So let us now look at the actual software and the databases which can be used in structure based drug design, and these are also important databases you must know for the CSIR net as well. So we have docking software like the Autodoc or the Autodoc Vena. This is used for soft docking and one of the most citing docking tool. It is fairly available, open source, and great for academic uses because it also important for the flexible docking. Another one is this Glint or Glide, which is used for Schrodinger. This is a commercial software and a gold standard. This has multiple modes like HTVS or high throughput virtual screening for fast rough screening. SP for stands for standard precision, XP extra precision, and FIFD for induced fit docking, the most accurate. Another one we have the dock, which is used for hard docking. This is the first docking program developed in 1982 by the Kearns, mainly used for hard docking and rigid docking. Good for the early screening of the molecules. We have Gold, which uses genetic algorithm and is very good for protein ligand interactions. This tool is also used for the soft docking. Apart from that, we have Flex for soft docking, and Fred is also for soft docking. So there are other databases we also have which we must remember. First is the PDB, one of the very important database, or we can say the database which contains 3D structure of the proteins. This is where you can download the target structures. Next is the PubChem, the largest collection of chemical molecules, and it is important because we can download the ligand for screening. Another one is the Zinc database, which is contains commercially available compounds important for virtual screening. Next is the Campbell, which is a chemical database, contains bioactivity data, how strongly drug binds to their targets. So these are the different databases and different tools which we can use in structure based drug designing. So once we identify the good drug candidate through docking, we need to check if they can actually work as a drug molecule or not. So they have to work inside the human body. That's why it is important to check the ADMET and their drug likeness properties. So what is ADMET? ADMET, where the A stands for absorption. Absorption means can the drug get from where we take it into the bloodstream, means how well the drug absorb into the bloodstream. D stands for distribution, means once in the blood can it reach the target organ or not. M stands for metabolism, which means how is the drug broken down, mainly in the liver. E stands for excretion, which means how is the drug removed from the body or excreted out. Last is the T, which stands for toxicity. Does the drug harm cells, organ, or cause any side effect inside the body or not? So this is ADMET. Now, a very important concept is the Lipinski rule of 5. So, Lipinski rule of 5 is also important from the CSIR point of view. Christopher Lipinski in 1997 studied orally available drugs and found that most of them follows 5 rules, where the rule number 1 is molecular weight. Weight must be less than or equal to 500. Next is the hydrogen bond donors. Hydrogen bond donor must be 5 or less than 5. Rule number 3 is hydrogen bond acceptor, which must be 10 or less than 10. The rule number 4 is log B, which is lipophilicity, must be 5 or less than 5. Next, the rule number 5 is rotable bonds, which should be 10 or less than 10. If a molecule violates any of them or more than one of these rules, it is likely to be a poor drug. It won't be absorbed well early. That's why it is important to follow the Lipinski rule of 5 by the drug molecule. Next, we have virtual screening. Virtual screening help us test millions of compounds computationally. Here, we use computers to rapidly screen millions of compounds. Those compounds are drug-like molecules against a target protein. So we identify the best drug candidate, and this screening has been done before any wet lab experiment. So look at the funnel here. We start with the millions of compound. First, we apply the Lipinski rule of 5. This removes the molecules that are too large and too polar. Then we do high throughput virtual screening, fast docking of approximately 50,000 compounds. Then we perform standard precision or standard precision docking on the top 500, then flexible docking on the top 500, and finally we get about 50 top hits that go for experimental testing. So as we know, the virtual screening is again of two types. One is the structure based virtual screening and another one is the ligand based virtual screening. In the structure based, we have the 3D structure of the target protein. On the other hand, we have in the ligand based virtual screening, we don't have 3D structure of the target protein. So this is the one major difference between these two type of screening. So in virtual screening, We can reduce wet lab cost approximately 95% by testing only top candidates, which saves enormous time and money in drug discovery process. So next, we have some real world examples of structure based drug designing. So let us now see all of them. Like first we have this Imatinib, which is used to target against BCR-ABL kinase enzyme. Here we have a disease called chronic myeloid leukemia. Now the structure based drug design was used to design a molecule that fit perfectly into the pocket of this enzyme and block the activity of this enzyme. So, in results, over 80% of the CML patients went into a complete remission, a revolution in the cancer treatment. So, another we have this Tamiflu, which is also known as oseltamivir in 1999. So, here the target site or the target protein is neuroaminidase enzyme. So, the 3D structure was solved, and structure based drug designing was used to design the drug against this influenza disease by targeting this enzyme, and Tamiflu prevent the virus from spreading. So this is the first line of antiviral against the flu. Next, we have Lopinavir or Ritonavir, which is developed in 1995 or 1991 to target the HIV protease. This molecule or the enzyme we use uh, to design the uh, structure based drug designing. And here, the crystal structure of this protein has been used. Now, the key component of HIV therapy. So here we used structure based drug designing to develop this uh, uh, medicine, or to develop this target associated drug molecule. Now last we have erlotinib, which is used to target this tyrosine kinase against the lung cancer disease. Here we have also used structure based drug designing, and this is a targeted therapy with fewer side effects, which is developed in 2004. So these example proves that the structure based drug design is a real working approach used in medicine today. So next we have some practice questions, like what in which molecular docking the protein structure changes as it shape to accommodate the ligand. So we have induced fit model, which we use in soft docking. Next, we what are the Lipinski rule of five? We have to select one which is not included in Lipinski rule of five. Next, we have which docking approach allows both protein side chain and ligand to be flexible in the soft docking, because this docking we also called flexible docking. The database is used to obtain the 3D structure is PDV, one of the important structure 3D structure database. Next, what stands for ADMET? Here T stands for toxicity, A stands for absorption, D stands for distribution, M stands for metabolism, E stands for excretion, and T for toxicity. So these are the few questions or the practice questions here we have. Now, lastly, here is the summary. So today we covered everything about structure-based drug design, how it differs from ligand-based drug design, role of protein 3D structure in drug design, workflow, binding site, different type of binding sites, molecular docking, hard and soft docking. Next, we have also studied induced fit theory and biological significance, the different tools used for molecular docking, what is ADMET and why it is important, what is Lipinski rule of five, what is virtual screening funnel, and next we have also studied the real drug examples And lastly, we have studied the different databases available to download the 3D structure of protein as well as the drug like molecule, which we call ligand as well. So thank you for watching.